So with that preface, I'll turn you've got the clicker already, over to Matt State, who's going to talk to us about genetics of autism spectrum disorder.
Thanks, Josh. I am going to talk today about genetics of autism, and I'll start just by letting you know that for several decades it's been clear that genes play the predominant role in the risk for autism. Not exclusive, but they do play the predominant role. But prior to the turn of the millennium, really there was very little progress, essentially no progress in identifying the genes that were predisposing or causing sort of common forms of autism. But by the late 1990s, an important observation was made, which was that children with autism were more likely to have gross chromosomal abnormalities, particularly new or de novo gross chromosomal abnormalities, than would be expected in the population. With the sequencing of the human genome and the development of high-throughput DNA assays, really the door opened to gene discovery in autism, and there was a seminal publication in about 2007 from Jonathan Sabat and Mike Wigler, where they used microarray technology to look at high resolution at the chromosomes of children with autism, and were particularly interested in families in which there was only a single affected child. And they asked the question whether or not there was an overrepresentation of submicroscopic changes in chromosomal structure in kids with autism versus controls. Those submicroscopic changes are called copy number variations, and indeed they found that copy number variations were more likely de novo to be found in children with autism than without. This served really as the inspiration for the development of something called the Simon Simplex collection, which was ultimately about 3,000 families, and it was the first large-scale effort in psychiatry to design a cohort that would maximize the opportunity to find new mutation. And that was done by the design you see here, where blue is unaffected, red is affected. The families that were recruited had unaffected parents. About 80% had unaffected siblings. There was always only a single affected member in the family. And that became the substrate for a series of studies using increasingly high-resolution methods to look for de novo mutations. I just want to point out that an important aspect of de novo mutation is that it occurs just prior to fertilization, not present then in the parents, not detectable in the parents, and is in every cell, germline de novo variation in the child. There's very little time for natural selection to act against those kinds of events. And consequently, you can have very large effect biological mutations that still are identifiable in offspring if you're able to detect them. I'm going to jump forward to 2014. That initial study from Wigler and Sabat was replicated by multiple laboratories, and increasingly the mechanisms to identify at higher and higher resolution changes in DNA became available. So something called whole exome sequencing began in about 2011, allowed us to look at every base of DNA in the coding genome. And what you can see from this figure here is that if we looked at a particular type of new mutation, those that were likely to affect the translation and transcription of proteins, that if they were predicted to be damaging, that we see an increased rate in children with autism, particularly versus their siblings. An important associated point is that when we look more broadly across the genome, the next panel shows actually whole genome sequencing, where most of the de novo mutations we find don't have that kind of impact on protein function. We see no difference between the rates in siblings versus probands. And similarly, when we look to see whether or not children with autism have multiple de novo mutations more commonly than their siblings, we don't see that either. So what this tells us is that this is not hypermutability. These are not random mutations through the genome. This is not an accumulation of multiple mutations. These are individual large effect mutations that are damaging to proteins, and those are the risk factors for autism. So we can leverage that to looking for repeated examples of the same gene or the same region being hit by these de novo mutations to begin to identify genes. This is a culmination of work. This was published in 2015. Stephen Sanders at the time was a postdoc in my lab. Now he's running his own lab at UCSF. It was a culmination of the work on the Simon Simplex collection, as well as all the other large autism consortium. We combine the work together. And what it shows you on the panel on your left are the chromosomal regions that were identified with, that are associated with autism. And then we can see approximately 65 genes that are reliably associated with autism, with this group here being the most stringent. We call them high confidence genes, and those meet the criteria for a whole genome significance, meaning you can correct for 20,000 genes in the genome, and we still see that they're associated. Now, we can leverage this information, begin to ask, what do these genes do? And very quickly, we see two areas of function that fall out, the synapse and chromatin modification and transcriptional regulation. Now, these are stunning to me to look at. I think some people have felt that it's a penetrating glimpse into the obvious. We have a neurodevelopmental disorder, and we've identified neural and development as being relevant. At the same time, though, even from these diagrams, you can begin to see that there is potentially traction around specific pathways and mechanisms that can be relevant for autism. Around 2011, as we were looking at these biological processes, Nanad Sestin, a close collaborator at Yale, and I began to think about whether or not there was a slightly different way to ask the question about autism genes. Instead of asking what are they doing, we wanted to know whether or not we could begin to identify when and where they were having their activity. So what we did was combine work in his lab that was looking at the transcriptional profiling of brain regions. So these are clusters of brain regions that were microdissected, and then he was looking at expression patterns. And these were done across all of development in postmortem brain from about 4 weeks of age in period 1 to 3 to about 80 years of age in periods 13 to 15. And we looked at clusters of gene expression, closely related genes, and across each one of the brain regions at each one of these periods in time. And we asked the question, is there any point in time and space in which autism genes are overrepresented in these? And in fact, we were able to identify that that was the case. So we could get spatial and temporal traction by looking at the combination of the genetic signature with the transcriptional signature. And what we found is that there was an enrichment of risk in the mid-fetal prefrontal cortex. We were able to get even more specific than that and identify that this was predominantly in deep layer cortical glutamatergic neurons. Now, Dan Geschwin's lab, this last paper here, they published in the same issue of Cell. They took a different, slightly different approach, a different gene list, but got essentially the same answer. Mid-fetal cortical development and glutamatergic neurons were critical for autism risk. So what I can tell you is that this look at de novo mutations transformed gene discovery in autism, both the structure and sequence of the DNA. These are rare, very rare in the population, but carry very large risks, orders of magnitude larger than we're used to seeing in something like genome-wide association. But genes are not fate. So these risks are large, but they're not specific. We see the same mutation in a child with autism can in another child increase the risk for schizophrenia, intellectual disability, specific language impairment. And clearly the question of what it is that takes these large genetic risks and canalizes them in such different ways is a critical question. So these large effect mutations have opened up really a new epoch in our ability to begin to gain traction on the biology of autism. But I can say that I think it also lays an important foundation to begin to ask what are the non-genetic factors that help shape either the course of autism, provide resilience, or take a single genetic mutation and have them in one case lead to one disorder and in another to something quite different. So thanks for your attention. I'm going to pass this on to
Thanks,
Chuck.
Matt.